Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about some basic principles of the process of transcription and translation. So this is meant to be like an overview of the genetic code. When I say transcription and translation, what I'm talking about is sort of the central dogma of molecular genetics, which is the transformation of material from DNA to its intermediate form RNA and then into a protein. So let's get into that conversation. And some of it may be a review to some of you who are watching this, but nevertheless, it's rather important. And so when we're talking about DNA, we're talking about this polynucleotide, deoxyribonucleic acid. And interestingly enough, along the sequence of nitrogen bases we're talking about, holds the recipe or the sequence for making a polypeptide. And sometimes multiple polypeptides will form into a complete protein. And so when I say the central dogma of molecular biology, it really means it. I really do mean it. So we're talking about DNA, and what's not shown in this picture is the intermediate molecule ribonucleic acid, and then that translates into protein. And so this is a fundamental and major concept of biology, and so it's certainly worth our conversation today. And so what I mean by transcription, what I'm getting at is the movement of the material from DNA into RNA. And so it's literally being transcribed into a different intermediate molecule RNA. When I mean translation, I mean this. The language of nucleic acid is being translated into a language of amino acids. In other words, the sequence of a polypeptide. And so this is meant to be an overview. And so let's get into that conversation. And so the material in DNA, the genetic instruction, we sometimes refer to as a gene. And so what is meant by that is a particular segment of DNA along this very large molecule. And so these units that we'd like to call genes provide the coding information to make a protein. And so they provide the, quote, instructions in the nitrogen base sequence for making the protein. Yet, it's not DNA that actually translates into a protein, rather it's RNA. And so what we need to talk about is how RNA is going to be the intermediate doing that. And so let's take a look. And so really, like this diagram, it pretty much says everything that you want to know about this. So this is obviously a eukaryotic cell. Here's the nucleus, and it's a nuclear envelope is a double phospholipid bilayer. Here are the nuclear pores. And so it's these nuclear pores, which are these gaping holes in the nuclear envelope that will allow RNA to exit from the nucleus. Now DNA is simply too large. It's too large to leave the nucleus, and for the most part you don't want it to leave the nucleus anyway in terms of regulating your protein production. RNA is rather clever that way because certain genes can be transcribed and then will make those proteins. And so if you think about it, if all the DNA were to go out into the cytoplasm, all the genes would be read and every cell would be making every protein, and that would be catastrophic. And so in terms of specialization in eukaryotic cells, it's good to keep your DNA back and use RNA. And then one of the cool things about RNA is that it's a short-lived molecule. And so there's RNases, which are enzymes in the cytoplasm that will destroy RNA. And so it only lives for a little bit of time. And so the news is current. So the cells only make proteins when they need to, and then the RNA is destroyed. And so transcription is basically how proteins are being regulated. And so that will be in a separate video, obviously. So this process of turning DNA into RNA is called transcription. Now this diagram, though I like it a lot, says that it's messenger RNA, but I wanted to point out that all the kinds of RNA are produced in transcription. And there's other types of RNA like ribosomal RNA, which make up the structure of a ribosome. There's transfer RNA that also is produced in the process of transcription that leaves the nucleus, and it helps to assist amino acids when they arrive at the ribosome. And then there's a whole slew of other RNAs. There's small nuclear RNAs, etc., interference RNA. And so all of the RNAs that are produced in the cell are produced in the same process in transcription. And that's catalyzed by a really important enzyme called RNA polymerase. And I'll just put it in right there. RNA polymerase. And that discussion of transcription will be in a separate video as well, because there's some detail which is very interesting in that process. So transcription. And then this little arrow right here is kind of humorous too, because not all the nitrogen-based sequences on DNA are coding, meaning they're expressed into the sequence of amino acids. There's a lot of the genes are interrupted by a lot of non-coding sequence called introns, and I'll write that out there. So these are non-coding sequences. And so before, now all of it's transcribed, but before messenger RNA leaves the nucleus, the intron regions, the non-coding regions, are edited and removed, sort of like a film that you need to process before the final production is released. And so a lot of the material, like a film, a lot of it is edited out. And so the messenger RNA that leaves the nucleus needs to be processed. And then there's a little something that happens to the 5' end of it. There's a guanine methyl cap that's placed on, and a polyadenine tail is placed on. But again, separate video. So that's called RNA processing. But let's just say that the RNA is produced and the RNA leaves the nucleus. And so that's one of the characteristics of RNA, that it can actually travel out of the nucleus. And so where does it go? It's going to a ribosome. And what's interesting is the ribosome is made up of a small subunit and a large subunit. And so the RNAs interact at the ribosome. So the ribosome is sort of the thoroughfare of the coming together of the transfer RNA, not shown, the ribosomal RNA, and the messenger RNA. And it's the messenger RNA that has the code on it in the form of nitrogen bases that are sticking out, which is what we want to talk about right now. These nitrogen base sequences is literally the genetic code for the amino acid sequence. And that's what we're attempting to do, is make a protein or a polypeptide. And so that's what we're talking about. And so the reason that this process is called protein synthesis is you're making a protein, but the reason it's called translation is because you're translating between languages here. There's a translation that needs to occur between the language of nucleic acids and polypeptides or amino acids. Find that very intriguing. So let's review a little bit, if this is a review for you, the structure of nucleic acids, because that's kind of essential in our understanding transcription and translation. As you might be aware of, is that nucleic acids are polymers of nucleotides. And so you're like, okay, so it's a repeating subunits of nucleotides. And this is what we mean by polymer. So many mers. So what's the mer? The mer is a nucleotide. And so what's a nucleotide? A nucleotide is what we'd like to call DNTPs. In other words, the N is replaced by either adenine, cytosine, guanine, or thymine. And so you have deoxyriboadenine triphosphate, or deoxyriboguanine triphosphate. So it's DATP, DCTP, DTTP, etc. And so what is a nucleotide? It's made up of the sugar deoxyribose, which is shown here. There's 3 phosphate groups, which remember are negatively charged. And there's a nitrogenous base. And we say nitrogenous base because it's rings with nitrogen contained inside of them. And so I want to point out that these DNTPs there are 4 of them that make up the nucleotide. But there is a difference in RNA and DNA. The nitrogen-based thymine is found only in DNA, and there's a separate nitrogen base called uracil that's found in RNA, but this is a better diagram of that. So here are the 3 nitrogen bases that are considered to be pyrimidine: cytosine, thymine, and uracil. Uracil is found just in the RNA molecule. So there it is, the nitrogen base. So you can see here, phosphate sugar base makes up a nucleotide. Now, the other 2 phosphates are not shown because those 2 phosphates are removed during the synthesis process. It's referred to as a pyrophosphate, and that provides the energy necessary for catalyzing the addition of the new nucleotide to the polynucleotide strand as it's growing. So these are the 3 pyrimidines, and then you have 2 purines, which are adenine and guanine. Purines because they're double nitrogen base rings. Okay, so this is a purine, this is a purine right there. And the pyrimidines would be, I did a different color, pyrimidines, we can say are the single ones right here. And so this one could be cytosine, this one could be thymine, this one could be thymine, this one could be thymine as well. So these are the single nitrogenous bases. Now if it's RNA, the thymine wouldn't be present, it would be uracil right there. And so that's what we're dealing with. And so I find this part to be interesting too. So do you see this pentagon in green right here? So the pentagon is a sugar. So it's a pentose, meaning that there's 5 carbons, 1, 2, 3, 4, and 5. And so those are named after carbon 1, 2, 3, 4, and 5, if you recall this from a previous video. So that's this sort of, the upper part of the molecule is what's considered to be 5 prime, and if you work it all the way down to this point, this is the 3 prime right there. So this is a polynucleotide, and each one of these is the nucleotide connected to the nucleotide connected to the nucleotide. And then of course here's a detailed drawing of the sugar deoxy. Now it's deoxy because, do you notice here on carbon 2 the hydroxyl group isn't present like it is normally in the sugar ribose. So ribose is found in RNA and uracil is found in RNA. And so that's the difference between RNA and DNA. Now, it's sometimes noted that most RNAs are single-stranded, and DNA is a double-stranded molecule that are held together by hydrogen bonds between the nitrogen bases. But the truth is, there are some forms of single-stranded DNA, and there's some forms of double-stranded RNA, so I don't want to get too caught up in that. So what I do want to say in this particular video is that in terms of how the genetic code is read, it's read in the sequence of nitrogen bases, because if I go back just for a second to the previous slide, notice this phosphate sugar, in other words, the side of the ladder, if you wanted to consider DNA or RNA as a ladder or a comb, whatever analogy you wanted. The only thing that will vary in the molecule is the sequence of nitrogen bases. So this could be A, and this could be T, C, A, T, T. It's the only thing that will vary because the side of the ladder is consistent. So the genetic code, it became clear to biologists that the genetic code is the sequence of nitrogen bases along DNA. And then ultimately, that same sequence of nitrogen bases needs to be transcribed into the messenger RNA, and that's unique, and that's going to be the recipe for proteins. And so genes are thousands of, or hundreds to thousands of nucleotides long, because proteins are very long as well in terms of their amino acids. So these genes, I'll just show it, which is again a segment of DNA, can be very, very, very long. Okay, and so they're found along the DNA, and so there's many possible combinations if you think about it. I know there's only 4 different nitrogen bases, but if you think about how long the genes are, it really creates quite a lot of variation in protein. And so ultimately, this is what we're talking about. The linear order of nitrogen bases in the gene specifies the order of amino acids along the polypeptide. And so if it's glycine or proline or threonine or glycine or lysine or proline or leucine or methionine, that particular primary sequence or the primary structure, in other words, the amino acid sequence of a protein, is very critical, because that ultimately will determine the type of folding that will occur in the secondary and tertiary and even ultimately the quaternary level of protein structure. So what DNA does, and then ultimately RNA, don't want to forget RNA. So the gene in RNA, in other words, the nitrogen base sequence of RNA will determine the amino acid sequence. Now, you may know already that there are 20 different amino acids found in living organisms. It's interesting, we can synthesize different kinds of amino acids, which is kind of clever, but there's only 20 found in humans, or not in humans, in all of life. And so these 20, well, the sequence will determine the ultimate folding. And so those 20 need to be spelled out. And how that's spelled out is the process of translation. In other words, how is the genetic code translated into an amino acid sequence is the question of the video. And so the primary structure, again, is important because it determines the three-dimensional conformation. And ultimately, the three-dimensional structure determines its functionality. And so that's rather cool. So if you're following this, you'll understand the fact that if you were to change the nitrogen-based sequence of DNA, you would ultimately change the protein amino acid sequence. And so the bridge between DNA and protein is, in fact, RNA. So you have to make RNA before you can make the protein, and that's called transcription. Now, of course, RNA is very similar. As we described, it has the same structure except for the sugar is different, ribose, and it has a different nitrogenous pyrimidine called uracil, which replaces thymine. It's often single-stranded, as I mentioned. And another fascinating concept about RNA is the fact that it's short-lived. The fact that that sugar, when it has both hydroxyl groups on carbon 2 and carbon 3, it allows RNA aces to degrade it rather rapidly in the cytoplasm. And so, as you can see in this picture, this is a picture of some sort of prokaryote, so perhaps a bacteria. Now notice there's no nucleus, and so it's a very simple single-celled organism. So the basic mechanism in a prokaryote, for which our understanding generally in biology, it came from, I find very fascinating, is that our understanding of the molecular basis of genetics comes a lot from microorganisms, and so prokaryotes are very, very useful. And so it's very simple. The mechanisms are similar between bacteria and our cells, or any eukaryotic cells. Of course, there's a lot more detail, but the basic mechanism is similar, meaning that transcription is the making of RNA, and then RNA will then be translated into a polypeptide. Now again, in a bacteria that lack a nucleus, transcription and translation are kind of coupled. So in other words, as soon as the message comes off of DNA, when I say message, I mean the RNA, as soon as it peels off of DNA, it's immediately picked up by the ribosome, and so proteins are being produced sort of simultaneously. So these processes are really linked together. But in the previous picture, if you remember, we talk about how that transcription occurs in the nucleus and translation occurs in the cytoplasm. But here in a bacteria, the ribosomes can start working on their polypeptide while transcription is still in progress, which is efficient. The bacteria need to crank out their proteins because a lot of them are short-lived. Before they divide, they need to be able to crank out proteins because they in ideal conditions, they're kind of dividing every 30 minutes. And so again, the process is very similar. Transcription occurs here, though, in the nucleus, and translation in the cytoplasm. And of course, there's some RNA processing that needs to occur. Why that doesn't need to occur in bacteria is because they don't have these intron regions, in other words, non-coding segments. And so they just simply, their RNA is pretty much good to go. But ours needs to be edited. And so transcription in the nucleus and translation in the cytoplasm. And so that primary transcript, if you can call it that, is then needs to be processed. So the introns need to be removed, and the cap needs to be put on the 5' in the poly A tail. And then it's exported. And so this is at the heart of really what I want to get at in this particular video, is how is the genetic code read? I'm going to create a separate video about how the genetic code was cracked. I find that to be even more interesting because, you know, when you read sometimes in a textbook, sometimes the textbook will simply just state what's happening. This is how the genetic code is read, and it's interesting. But I want to make it clear to everyone who's watching that the textbook is really the culmination of the work of a scientist, which is really discovering things through experimentation. And I think the cool stuff about science is how did the scientists come up with it? I mean, there needs to be an experiment that shows the information, and so that's really important. But this is just simply, this is how the genetic code is read. And so one of the dilemmas about reading the genetic code was, you know, how does this work? If you have something like this, these nitrogen bases in RNA, and you're trying to do amino acids over here, the cell had a pool of amino acids, and say there's amino acid 1 and 2 and then 3, and these little flags represent the R group, and so there's 20 of these. Let me just jump ahead to 20. There's 20 different ones, and so how do you decipher or translate the genetic code? It seems interesting to me because if adenine coded for one of the amino acids, that's great. And uracil coded for another one, and cytosine, and then guanine. You're like, this is great, but there wouldn't be enough. So single nucleotides wouldn't be enough to code for all 20 of them, because you'd run out of names. It's like 20 people and there's 4 names. You're like, well, perhaps the genetic code is read in doublets. In other words, little groups or pairs. So AU, CG, CC, like that. And so if you did the math, if you said if it's read in groups of 1, there'd be 4, and if you read them in twos, it'd be 16 combination. It's not enough. So the genetic code is actually a triplet codon. And so each of the 3 nitrogen bases, 1, 2, 3, 1, 2, 3, 1, 2, 3, and 1, 2, 3. Each of these 3 code for an amino acid, an amino acid, an amino acid, and an amino acid. So the genetic code is a triplet. And so the code, each of the 3 nitrogen bases is referred to as a codon. A codon. So that's how the genetic code is read. So they're triplets of nucleotides. I thought this was funny. Triplet bicycle right here. So that's what codes all of the amino acids. So the triplet code, 3 consecutive bases in order. They're linear. They need to be read in a certain orientation. I'll mention that in a moment, from left to right. And also, when you think about it, if it's read if there's 4 nitrogen bases and you read them in groups of 3, that's going to create 64 possible word combinations. Now that's over what you need because there's only 20 amino acids, but that's okay, because as it turns out, there can be changes in that nitrogen base sequence called a mutation, and it still may result, because of the redundancy of the genetic code, since we've overdone it, it'll still result in the same amino acid, and so that's fairly helpful for the cell. So the genetic instructions are written out ultimately in the form of a polypeptide chain, but the instructions are written in 3-letter words in DNA, and so this is a great diagram of that. So here's a DNA. This light blue might represent an intron and non-coding regions, and then here's a gene. So a gene is a segment of DNA that's rather long that encodes for a polypeptide. And then the genes are often interrupted in a eukaryotic cell. In a prokaryotic cell, the genes flank each other for the most part, although there's more detail to that. Now, I wanted to point out something that's rather important, because the correct orientation of the nitrogen base sequence is rather critical. And so you might know that DNA is a double-stranded molecule. There's 2 strands. And so as it turns out, the 3' to 5' is the sense strand, in other words, the template. That's really important, because when you transcribe it into RNA, so do you notice here A bonds with U, C bonds with G, C bonds with G, and this spells out a codon. And then AAA is then transcribed into UUU, and then CCG is GGC, like that. It's very important, the orientation, because obviously, if you're following this, it's different if you spelled something UGG than if you spelled it GGU. So the orientation is important. So the genetic code, and I just want to emphasize this, I'll put a little asterisk. The genetic code in messenger RNA is read from the start of the 5' side. Okay, so there's a 5', and when I'm talking about 5', I mean the phosphate group over there, and then this is the bottom of the sugar over here, this adenine. So it's read in that orientation from 5' to 3'. And so when I say 3 makes up an amino acid, this is what I mean. UGG codes for trip, and UUU is phenylalanine, and GGC is glycine, and UCA is serine. So this is how the genetic code is read. And so there's a template strand, and you're like, well, what's the other one? The other one's complementary strand. And so that's important in terms of DNA sequence. Of course, they're complementary, but one of them contains the sense strand, if you will, to produce the messenger RNA, which is then translated into the protein. Now, just to back it up a bit, all this genetic material, in other words, the nucleic acids of DNA, are coiled very tightly around these proteins called histones, forming these structures called nucleosomes, in other words, like string along the beads. And as you may be familiar with, these really condense and supercoil, these nucleosomes do, into chromosomes at the onset of a process called mitosis. So chromosomes are formed during prophase of mitosis. But if you were to uncoil that DNA from the histones, you can see the double helix here. And so what we were talking about is a gene right here, which is a sequence of DNA that codes for a polypeptide. And again, this is what we're talking about. The gene is up here, and then it codes into a polypeptide. And so during translation, groups of 3 code for the sequence of amino acids. And so as I was emphasizing before, translation, it's read from the 5' direction to the 3' direction. That's important, or else you'd be spelling it backwards. And so if you think about it, if there's 3 nitrogen bases for every 1 amino acid in a polypeptide, it'll take 3 times the amount of nitrogen bases for the amount of amino acids. So it would take at least 300 nucleotides to make a polypeptide that was 100 amino acids in length. And if you follow that, then that means that you're understanding the point of the video. And so genes are then transcribed into messenger RNA and then translated into proteins. And so I hope you enjoyed that basic overview of the process of transcription and translation and reading the genetic code. I hope you're so intrigued that you'll want to watch future videos on cracking the genetic code and the details of transcription and translation. Again, thanks for watching.